Good evening to all of you. My voice is audible. Please let me know. Any one of you, my voice is audible. Good evening to all. How are you all? Please let me know. My voice is audible. Okay, okay. Yeah. Good evening. Good evening, Rihanna. Good evening. How are you? Muskan. Good evening. Yes, Ankita. How are you all? Everyone is fine. Just let me know. My voice is audible. Okay, okay. You all are fine. Okay, good, good. How is your day today? Have you, how is your day today? Okay, okay, okay. Thank you all. We have Muskan, Pritam, Harshini, Jagriti, Muskan, Shivam, Rihanna, Ankita, Nilima, Soumya, Chandrala. So have you done the homework also, which you have to do, or you will take one more day because we have done little bit extra yesterday. So I think at least 2 days should be there for all of you. Udita, it's your wish if you want to write it by your own hands or by copy or pen, or if you want to submit by MS Word, it's your wish only. How you are comfortable, according to which you have to do it. Okay. So no any regulation from my side. It's your wish only how you are feeling comfortable. So I just want to do the topics little bit fast so that you will be free by 15th of December, and after it you will be focused on your exams as well as on your review paper. So in the meantime I will not force any of you to submit the review paper or something, but yes, you have to send me your assignments on time so that you will be able to get the certificates easily. Yes, yes, I am checking the abstracts. I am checking it, I am working on it. No worries regarding it. Do you have any another query? Please let me know. Because right now I am not forcing any of you regarding the abstract or review paper. Just complete the assignments on time. By after 15th of December, you have to focus on the review paper. Yeah, hello Ritu, how are you? Yeah. No worries, no worries. You don't do it by midnight. Try to submit it by tomorrow. Okay. Don't be too much. Don't feel so much stressed for these assignments. Take your time. Kindly submit it by tomorrow also. No worries regarding it. No, 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 not everyone has to mail. Only one has to send me the abstract only, along with the name of other persons who are there with you for this review paper. So only one person will send me the mail, but in the abstract in the topic, right after topic, you have to add the name of all of those who are involving in that particular review paper. I get the review paper from many of you. Just give me some time. By this weekend, by Sunday or Saturdays, I will be free from the these lecture series, and then I have ample of time for all of you to read your abstracts, your topics with full concentration. Because right now my schedule is very busy. From 4 to 10 p.m. I have my continuously lecture series, so I'm not able to check it properly. So just give me the time up to this weekend. I will definitely check it and mail you as soon as possible. Okay. I hope you all will understand. But in the meantime, you all have to try to think of your topic. You have to write the abstract too. Dipti have not enrolled for bioinformatics. Dipti, I'm sorry, I'm not getting you. What is basically you want to tell me? I'm not getting you. Please elaborate it more, please, if possible. Yes, Imran, good evening. Shivam is here. Yeah, Shivam, just give me a little bit more time. I will mail you as soon as possible. Ishita is here. Good evening to all of you. Just waiting for 1 or 2 minutes more so that students will join and then we will start the session. And I will try to complete the topics so that you will be free by 15th of December from my side. Okay, I will try to. No worries, Juvin. Take your time. Just send it me by tomorrow. Okay, Soumya. Okay, I will check it. But I will reply to any one of you. So you have to coordinate with each one. I will reply to any one of you, and then you have to let us know to each other. Okay. Okay, Shivam, okay, submit it by tomorrow. But please, please, assignments are very much important because you have the regular assignments up to 15th of December. After it, your lecture series will be wind up, and you will have to focus on the review paper then only. But for these 5 or 6 days, you must have to submit me your assignments on time. I will try to complete or wrap up the lecture series by this weekend, by Monday or Tuesday definitely. So you have to keep your assignments on time. Yeah, yeah, please try to limit your abstract by 200 to 300 words only. You can write as much as you want to write in the review paper, that means in the full paper, but abstract should be limited to 200 to 300. Otherwise, the journal will not accept it. Okay, so you can write as much as in the review paper. Instead of 5 to 6 pages, you can take it up to 10 pages also. But for the abstract, I am very sorry, it should not be exceeded by 300 words. Yes, Pragya, I will check it. No worries. Okay, okay, Soumya, I will definitely go through it. Don't worry. So I request all of you again. I know you have your busy schedule also. So it's okay if you are not be there live on the classes. It's okay if you have some exams or something. But it's my request to all of you to submit your assignments on time because I have to keep the record of that particular thing. I have to check it on that particular date. So please try to submit me your assignments on time so that you will have not any problem during your certificates. Okay, Swapnil, okay, good. Just send me your assignment. Jagriti, you send me a complete review paper. Instead of abstract, you send me complete review paper. Very good. I will check it. No worries, I will check. So those who are in the internship, uh, okay, Shruti, I will reply just right after this class. I will definitely reply. So there is only one thing which I want to let you know that for internship review paper is compulsory. Otherwise, those who are not in internship they can even skip it. But if they want to learn something new, they are most welcome. They can write it as well. So I think we should start now. We have just waited for few minutes to the students, but they will watch it later if they are not present right here. So now I am going to start the session. So this is the syllabus which is we have along with us. Yeah. So we have done this recombinant DNA technology. We have covered separation of molecules by electrophoresis. We have also completed this quantification, separation and purification. For separation is with the help of electrophoresis, separation is with the help of this chromatography also. Quantification is basically separation and quantification are quite related with each other. So we have discussed all these things in the proteins PPT, which I will share with all of you on today. Right after this lecture series, I will try to share the PPTs also. Now we have another term which is associated with this protein part, which is the blotting techniques. These blotting techniques is also the techniques of MBBT. So these will be covered in these molecular biologic techniques also. Otherwise, we will focus on the blotting techniques which are there. After it, we have microscopic part which is left. Microscopic part is there. Some applications of recombinant DNA technology. Also, I share the PPT of recombinant technology with all of you. But my main focus, right after this blotting technique, my main focus will be on microscopy. Plus, I want to do this thing, which is flow cytometry. So these flow cytometry and microscopy will be there after the blotting techniques, which are very much important and interesting also, so that you will get lots of knowledge regarding microscopy and flow cytometry. This we have already done on the very first day. So we have just left with the after it we will just left with the techniques which are there, the techniques which we will try to cover even today also. So, but the main thing which left right now is the blotting techniques, flow cytometry and microscopy. Okay, rest are only the applications, and yes, bioinformatics also. So bioinformatics is another field only. I also take the bioinformatics batch in the previous month also, around Diwali. I have completed a bioinformatics batch also. So if you want to go through those videos, you can go through those videos also related to bioinformatics. Okay, but in this lecture series, we will limited it up to only 1 day or 1 hour. Otherwise, there is totally a 20-hour long session on bioinformatics also, which is available on my channel. So if you are interested in bioinformatics, you can watch those videos also. Otherwise, for this lecture series, it is only for 1 hour. So our main focus will be on blotting techniques, flow cytometry, microscopy and bioinformatics. So are you getting it? Are you comfortable? Soumya, the network is quite good here on my side. I will suggest you to kindly refresh it, so that maybe because of network issue you are not getting it right now. I just suggest you to go to refresh your YouTube, please. So we have to start with blotting right now. So are you getting me? My voice is audible to all of you. Please let me know. So we will try to complete blotting techniques today. After it, we will have to complete flow cytometry, microscopy, and bioinformatics. Then these applications part is very simple and you will get it. These are the some new things which will be new to all of you, maybe for some of you. So now I am just starting with the blotting. So this was the little bit history, because it's very important. We must know from when and from where these techniques come into existence. So it was around in 1963 when we were able to use electrophoresis, when we will able to use nitrocellulose filters which are there in the blotting techniques. I will show you later in the later slides. Then in the 1966 and in 1605 we have some scientists who discovered that nucleic acid fixed can be detected with the electricity sensitivity by hybridization. Yes. So there is one more term which will be there which is hybridization, and one more term is there which is radio labeled probes. So these terms will be quite common in the plotting technique. In 1970s, the possibility of mapping whole genome arose along with the era of rDNA and gene cloning. Thus, need to find a single gene among the thousands of fragments of a DNA was needed. So they are just letting us know that why there is need of separation of a DNA, why we have to focus on a single gene. Because when we are doing the genome mapping, at that time it is needed that we will work on a single gene, or we must know the functioning of each and every gene. So that is why the at that time in 1970s it becomes a need to separate the DNA molecule. Then after it in 1975, this is the scientist Edward Southern. From this year we able to know about some blotting techniques. So in 1975 we will come along with southern blotting. Then in 1977, 2 years later, we will get northern blotting. In 1979, this was a scientist talk who developed the protein blotting, or you can say western blotting. So the very first was in the year 1963, when we are able to use the nitrocellulose membranes, the nitrocellulose filters, but only in 1970s during genome mapping we came to know about the need to separate the genes. Then in 1975 southern blotting, 1977 northern blotting. I hope many of you are much aware of it, but still I keep it very simple. What is basically blotting? So visualization of a specific DNA, RNA and protein among many thousands of contaminating molecules. So when we specifying a particular thing, when we are just focusing on a particular biomolecule, suppose I am focusing on a DNA, so the all other things which are there, the RNA, the proteins, they will be contamination for me at that time. So visualization of a specific DNA, RNA and protein among many thousands of contaminating molecules requires the convergence of number of techniques which are collectively termed as blot transfer. So basically this is blotting, when we are able to visualize a particular biomolecule, biomolecule. So one more term is there which is known as hybridization. Now what is basically hybridization? Hybridization is basically formation of a stable base pair between the probe and the target DNA. So any one of you please let me know what is probe, so that I will came to know how much you know. What is probe basically? Any one of you? What is basically a probe? Or you can say, what is basically hybridization? Any one of you? Please let me know so that I will able to know that whether you know about blotting or you don't, you are new to it. Probe. Please let me know about DNA hybridization. Any one of you, not anyone but all of you, please let me know about DNA hybridization and about probe. Yes, yes, it's a soft complementary DNA or RNA sequences. Okay, good. Pallavi is probe is basically sequence of nucleotides. Okay, good Pallavi. It is complementary to the target sequence. Okay, Asim is probe is a DNA fragment that is used to hybridize DNA In nitrocellular crystal. Okay. Anal is small piece of a DNA. It's which has some integrating property. Okay, very good. Basically, what is hybridization? What is hybridization? Shruti is so Muskan, DNA hybridization is used to determine the degree of similarity between different DNA sequences. A probe is a single-stranded sequence of DNA and RNA used to search for its complementary sequences. Small piece of DNA or RNA which is radio labeled. Very good, Nancy. Yeah, the probe is basically radio labeled. Shruti, probe is labeled with a radioactive substance or a chemical. Here, probe is a single-stranded DNA which is used to search complementary sequences. Very good. Priya. Monogenes DNA hybridization generally refers to molecular biology technique. Yes, so it's a part of a molecular biology technique that measures the degree of genetic similarity between the pools of data sequence. So I came to know that all of you have ample of knowledge regarding this hybridization and blotting. So it will be easy for me to then discuss it. Muskan is hybridization is the process of combining two complementary single-stranded DNA and RNA and aligning them to form a single double-stranded molecule. Sangeeta, hybridization generally refers to molecular biology technique that measures the degree of genetic similarity between the pools of DNA. Okay, okay. So thank you all for your replies. Thank you Priya, Ichita. Thank you Enal, all of you. So yes, you are aware of these terms. So hybridization probe is a fragment of a DNA or a RNA by variable length which can be radioactively or fluorescently labeled. So this blotting is definitely a part of this molecular biology technique. So when we are covering this blotting, it will also cover in this molecular biology techniques part 1. Okay. So yes, thank you all. So let me continue it now. Okay. So what is now hybridization basically is the formation of a stable base pairs between the probe and the target DNA. So we have a target sequence, we have a target DNA with which we will allow the probe DNA which is generally radio labeled. We will allow this DNA, this probe DNA which has a complementary sequences along with the target sequence to bind and they will form the hybrid. They will form the hybrid. So probe which is radio labeled, they will bind with the target sequence and they will form the hybrid. So now what is basically blotting? So blotting is the technique for transferring DNA, RNA and proteins onto a carrier. So it is not, blotting is basically not for the separation. Like electrophoresis is the separation of the DNA, RNA and proteins. But blotting is not the separation. Rather it is a transferring. It is a transferring of the DNA, RNA and proteins onto a carrier. So these DNA, RNA and protein which are present in the gel, we have the bands here, they will have to transfer onto a carrier so they can be separated and often follows the use of gel electrophoresis. By the use of gel electrophoresis, we will able to separate the DNA, RNA or the protein or you can see the biomolecules. But after the separation, we have to transfer them. So blotting will come right after the electrophoresis or you can say right after the separation we have the transfer. We have to transfer those DNA, RNA or protein. So this is known as blotting. So blotting is a method of putting DNA, RNA or proteins onto a membrane for further studies and detection. So it is basically transfer. We have to transfer them onto the membrane for further studies and detection. So we have three types of blotting. We have three types of transfer. The number one is sudden blotting in which DNA is detected with a hybridization DNA or a probe. So in the sudden blotting, we are basically focused on the DNA. We have to transfer the DNA and it is generally done with the help of a probe. Similarly, in case of northern blotting, RNA is detected with a hybridization DNA or RNA probe. In case of western blotting, here proteins is detected with a complementary antibody. So in case of RNA and DNA, we are using the probes for hybridization, but in case of western blotting, we will follow the antibodies. So the three blotting techniques have similar methodology. So these techniques are similar, only the biomolecules will be different. So the three blotting techniques have similar methodology. Molecules are separated by electrophoresis and are transferred to the membrane by blotting. That is specifically suited to score the detection of a fragment with a particular DNA sequence, single species of RNA or protein. So basically, along with electrophoresis, we have to do the transfer to the membrane. So number one is southern blotting. It was developed in 1975 by the scientist whose name is Edwin Southern, and it was basically used to detect the DNA. So this southern blotting is basically for the detection of the DNA. So this method involves separation. Now how we will have to do the separation? With the help of electrophoresis. Separation, and then we have transfer, after it we have hybridization. Hybridization is which? What is basically hybridization? When we are allowing the probe to bind with the target DNA sequence. So the DNA which we have to detect, it can be a single gene or it can be a part of a larger piece of a DNA which may be a viral genome. So this is basically southern blotting. There is three steps: separation, which is number one, transfer number two, and hybridization is number three. So what is the principle of this southern blotting? The mixture of molecule is separated, immobilized on a matrix, probe addition to the matrix to bind to the molecule, unbound the protein. So first of all, we have to do the separation by electrophoresis. Number two, we have to transfer it on the nitrocellulose membrane. Number three, we have to add the probe. And number four, we have to wash out the unwanted probes. Getting my point? So these are the steps which are there in the southern blotting. Number one, the DNA is digested. After it we will get the fragment. So after DNA isolation, we will get the DNA which we have to run on gel electrophoresis, which we have done already. After this separation, we have to do the transfer. So after gel electrophoresis, they will be transferred to the membrane. After it, probing is there. At the end, we have to watch it under the autoradiogram. So these are the steps which are involved. The main three steps are separation, transfer and hybridization. So these are the three main steps in the case of blotting. We have separation, we have transfer, we have hybridization. So this is the first step of a blotting to be discovered, which plays a significant role in recombinant DNA technology as well as in molecular biology. So what we have to do, we have different types of biomolecules, but we just want a specific biomolecule, which may be a DNA or RNA or a protein. So we just want a target DNA on which we want to work further. So the number one thing will be the separation of the molecules. So this is again the preparation of the sample and running the agar gel. Then after it we have to transfer it on a nitrocellulose membrane. Probe will be added. Probe will consist of pre-hybridization, hybridization, post-hybridization washing, and after it we have to watch it under the autoradiogram. So probe with the hybridization has three steps basically: probe preparation, pre-hybridization and hybridization. So, and also the post-hybridization, which is the movement of the excessive probe. Now this is the sandwich which we have to make during the transfer of the biomolecule. So the sandwich consists of a filter paper. The sandwich consists of a filter paper. On it we have the nitrocellulose membrane. On it we have the gel matrix, and on it we have the another gel paper. Sorry, filter paper. Let me show you. So if you are able to see here, this is a gel which we will get after the electrophoresis. Suppose this is a gel which we will get, in which we have a band, like here we have a band. Now we have to transfer from this electrophoresis, from this gel which is we have just do the electrophoresis, we have to transfer it into the nitrocellulose membrane. So DNA fragments which are separated by electrophoresis, now they will have to pass on to the or transfer onto the nitrocellulose membrane. So what we have to do, we have to put up our apparatus, we have to separate the DNA fragment and those paper. So what we will do, we will use alkaline solutions, we will just keep the agarose gel. Above the agarose gel we have the nitrocellulose paper, and above it we have some little bit of weight also. So what will happen, the gel, the all the bands which are there in the gel, they will come out on the nitrocellulose membrane. So this membrane, there is a transfer of the bands from the gel to the membrane. Now we have to do the next step, which is the adding of the DNA probe to separate DNA. So by hybridization, by adding the probes, we get the desired proteins, or you can say desired DNA, which will be labeled, because probes will be specific to a particular sequence. So they will, excessive probes will be washouts, and we will get the bands. So labeled DNA probe hybridized to complementary DNA bands visualized by autoradiography. So this is the basic thing, the basic phenomena of blotting. So you have to remind only 3 points. We have separation by electrophoresis, after it we have transfer, after it we have the hybridization. Only these three things are there in case of blotting. If you understand this basic thing, then blotting is nothing for any of you. Even this blotting technique is quite common these days in our molecular biological labs. If anyone, maybe the scholar or any one of you, must be have done these techniques in your labs also, and even there is workshop for these techniques, for this SDS technique, for this Western blotting, for this sandwich model. So we have workshops also for 2 or 3 days, so that you will be able to get the practical knowledge, because right here I am only able to give you the theoretical knowledge. I will only let you know about the basics of blotting, the steps which are there in the blotting, but you will get the knowledge only by doing it practically. Getting my point? So all these things are practically, you have to do it practically, but till before doing practically, your theoretical part should be strong. So you have to keep these three things in mind, that blotting consists of separation by electrophoresis. Number two, we have to do the transfer of these bands from this gel to the nitrocellulose membrane. Number, and the step will be hybridization. This is a very basic step which is there in the southern blotting. Now just let me check everything is fine. So are you getting me right now? Are you getting me? Are you getting me? Hello, my voice is audible. Are you getting me? Any one of you, please reply. So that I will continue it further. Please let me reply. Oh, okay. Just we will wait for 1 or 2 minutes so that it will go okay. Then we will start it. Otherwise, you will able to understand the phenomena which is there in case of blotting. The main three steps which is there in the blotting, we have the three main steps, which is number one is separation, number two is transfer, and number three is the hybridization. Are you getting it or not? So you have to keep only two things in mind, that electrophoresis is the separation of the proteins, and after the separation of the proteins we have to do the after the separation of proteins we have to transfer them to the nitrocellulose membrane, and after the transfer it onto the nitrocellulose membrane. Okay. After transferring it into the nitrocellulose membrane, another step which is there is the hybridization. So the next step is basically the hybridization, when we have to add the probes. When we have to add the probes, and the probes will bind to the specific DNA sequences. They will, yes. I'm just waiting. I'm not just going further. Let me check it when everything is fine. Then I will start only. No worries, please don't worry. If everything will be fine, then I will continue. Okay. In the meantime, I am just letting you know about the main things which are there in the main thing which is there in the blotting. So you must know the critical things which are important in case of blotting. So the three steps which are there are: number one is the separation, number two is the hybridization, number two is the transfer, and number three is the hybridization. Okay, just I am waiting for 1 or 2 minutes, so that if the video is not buffering now, then I will start only. Okay, just waiting for 1 or 2 minutes more. I think it's not buffering right now, so I think I should have to continue. So may I continue now? Okay, thank you. I think it's okay now. So yes, so this is the basically southern blotting which was there in 1975. The scientists southern developed this technique that is widely used to detect the fragments of DNA. Now these techniques require the separation of DNA by electrophoresis. We have discussed it. DNA fragments are blotted on a strip of nitrocellulose, that means transferred. Now we have two things which are there. We have nitrocellulose membrane as well as we have nylon membrane. Now this is the homework for all of you for today. Please, any one of you, please write it somewhere, that why do we need nitrocellulose membrane as well as nylon membrane. If nitrocellulose membrane is okay, why there is a need of a nylon membrane in case of this transfer of the proteins or the biomolecules? Why there is a need of nitrocellulose membrane, or you can say nitrocellulose paper, as well as the nylon membrane. So you have to let me know why these two are needed simultaneously. And the third step is identification by hybridization with a labeled probe. So are you able to get the question? Let me check it. Are you able to get the question? Are you able to get the question? So you have to let me know uh, if there is nitrocellulose membrane or nitrocellulose paper is there, why there is a need of a nylon paper. Okay, if there is a nitrocellulose membrane, why there is a need of nylon paper. This is your homework for today. So are you all getting the question? Okay, so any one of you please write somewhere, and then you have to upload it in the group also. Any one of you please, you have to pin it later on also. So please, yes, yes, you will. So yeah, why do we have a need of nylon as well as the nitrocellulose paper? So write it somewhere so that you will share with your friends also. Now, these are the steps which are basically there in case of southern blotting. So, southern blotting, a method for transferring the DNA from an agarose gel into a nitrocellulose filter on which the DNA can be detected. The DNA, now this is a procedure how we have to do this whole things. The DNA sample is digested by restriction endonucleases, producing small fragments and that are amenable for analysis. Fragments are separated by agarose gel electrophoresis or by page. So we have to do SDS page later on, but just keep it in mind that a page and electrophoresis is basically for the separation only. The mobility of nucleic acids in agarose gel is influenced by agarose concentration, molecular size and molecular conformation of the nucleic acids. Agarose concentration can be 0.3 to 2%. It is the most effective concentration for the separation of the nucleic acids. So like proteins, nucleic acids migrate at a rate that is inversely proportional to the logarithm of their molecular weight. So separated molecules are visualized by etherium bromide. So this is everything which is there in case of electrophoresis. So I am not going to discuss it again. Then electrophoresis can be performed with the dye in the gel and buffers, and we have to watch this gel under the, we have to observe this gel under the UV light. Basically we have to watch it under the UV light to check the color. So during UV light or during using etherium bromide, you have to put the gloves and everything is there. But our main focus is transferring, because the rest is this thing is basically electrophoresis, but main focus is transferring. So transfer of the DNA from gel into nitrocellulose membrane done by weak acid treatment to defluorinate and fragment the DNA, thus make it smaller and easier to elute from the gel, followed by denature with strong base and neutralization. Single strands bind to the membrane more efficiently. A buffer is used to facilitate the transfer. So when we want to transfer the bands from the gel to the membrane, lots of things are keeping in mind. We have to use a particular buffer also to facilitate the transfer. So original methods of transfer relied on capillary action basically. So how we will do the transferring? By capillary action only. Vacuum or pressure system can be used to speed the transfer. Faster and more efficient transfer is afforded by the use of electroblotter. Electroblotting process is usually complete in 14 hours. So basically for the purpose of transfer we have to use the capillary action. Basically we are working with the capillary action, but sometimes for the fast processing we are able to use electroblotting technique or electroblotting process also. So this is gel electrophoresis, this is again electrophoresis. Please read it by your own self. Now this is the thing, we have to transfer the DNA from the gel to the nitrocellulose membrane. Now we have nitrocellulose membrane, we have a nylon membrane. So they have some characteristic features, that is why we are using both of these. But you have to tell me the features, okay? I will not share right now. So this is a gel where we have the bands. By electrophoresis we will get the bands. Now we have to transfer these bands. In the pros we will visualize it under the autoradiogram. The same thing is repeating again and again. Now this is the difference between nitrocellulose membrane and nylon membrane. Why we have nitrocellulose paper as well as nylon membrane for the transfer of the bands only. So this is the transfer method, and you have to keep in mind the transfer is by the upward capillary action. So by capillary action the band will slowly, it will take lots of time, slowly and slowly they will transfer from the gel to the nitrocellulose membrane. So rate of a transfer depends on the size of a DNA and concentration of a gel. So if you one is the gel, so this is the gel. After it, we will have to place the nitrocellulose membrane. So just right after gel, this blue line, this is the nylon paper. After it, we have to put Wattman filter paper, we have to put another paper, and we also have to put a weight, so that the process of capillary action should be little bit fast. Okay. So this is the transfer method. This is basically done with the help of upward capillary action. Please let me check this. Is it okay now? Or is it? So this is the whole gel apparatus which I just share with you right now. That we have this yellow one which is the gel, above it the blue one is the nylon membrane, above it we are just placing the filter papers, the paper towels, the weight is also there, because this works on the capillary action, upward capillary action. From the gel the bands will transfer to the membrane. This is the downward capillary action. Sometimes we are using downward capillary action also. Sometimes what is there, simultaneously transfer to two membranes instead of one. We are able to transfer the gel, one membrane is above and one membrane is below. So from one gel we are transferring it on the two modifications of blotting basically, but this is the basic technique of transferring only. Otherwise, we have downward capillary action also, as well as we have the simultaneous capillary action also. Now, this is the electrophoretic transfer. This is again another method for the transfer of the proteins or the biomolecules from the DNA gel, of the proteins, biomolecules or the DNA from the gel to the nylon membrane. Vacuum transfer is also there. So these are little bit techniques which are there in the blotting. Now the third step, which is quite important, which is known as hybridization.